All right, let's dive right in. Today, we're going to explore the absolutely fascinating world of bacterial gene regulation. Now, this isn't just about a few simple on-off switches. We're talking about a super sophisticated, multi-layered network that is the key to how bacteria not only survive, but completely dominate some of the most extreme environments on Earth. So let's start with the most basic question of all: why?
Okay, so why do bacteria need to bother with all this complex regulation? I mean, how do these seemingly simple, single-celled organisms manage to set up shop in pretty much every corner of our planet? Well, the secret, the real magic, is in their incredible ability to listen to their environment and respond with unbelievable speed and precision.
And when I say every corner, I really mean it. Just look at these examples. We're finding bacteria thriving in boiling hydrothermal vents, in the crushing pressure of the deep sea, and yes, even living inside our own brains. Their trick isn't having a huge, unwieldy genome. No, their genius lies in having a dynamic, flexible genome and being absolute masters at controlling which genes are on and which are off at any given moment. That is the secret sauce to their survival.
So you see, for a bacterium, this whole gene regulation thing isn't a luxury. It's a matter of life and death. It's what allows them to pivot in a split second when a new food source shows up, or a stress like an antibiotic appears. It's about being incredibly efficient, not wasting a single molecule of ATP building a protein it doesn't need right now. It all boils down to adapting, optimizing, and outcompeting everyone else. It is survival of the fittest, played out in real time at the genetic level.
This relentless pressure to be fast and efficient has literally shaped the way the bacterial genome is structured. It's not just a messy library of genes. It's organized with an almost ruthless logic. So let's unpack the key design principles that make this whole system possible.
The most fundamental organizational trick is the operon. The best way to think about it is like a dedicated toolkit for a specific job. Let's say a bacterium wants to break down lactose. It needs a few different enzymes to do that. Instead of having 3 separate on switches for 3 genes, the operon bundles all the genes for that one task together under one single switch, the promoter. You flip that one switch and you get the entire protein toolkit all at once. It's just beautifully efficient.
Now, if we zoom out a level, we get to the regulon. So if an operon is the toolkit, the regulon is kind of like the master contractor overseeing multiple projects. It's a single regulatory protein that acts as a master switch, controlling many different operons and genes, even if they're scattered all over the chromosome. This allows for a massive coordinated response. A single stress signal, like sudden heat shock, can trigger one master regulator, which then activates a whole army of different toolkits needed to fight back and protect the cell.
And this map of the E. coli chromosome really brings these ideas to life. You can actually see some of the famous operons you've no doubt studied. There's the lac operon for using lactose, sitting at about 1 o'clock. You've got the trip operon for making tryptophan down at 4 o'clock, and the his operon around 9. Each one is a neat, self-contained unit, but a single master protein can coordinate their activity as part of a much larger regulon, creating this beautiful, integrated genetic network.
Okay, so with that genomic layout in mind, let's drill down into the very first layer of control, transcription. This is the moment the cell has to decide, are we even going to start making this protein? From an energy-saving perspective, this is, without a doubt, the most important checkpoint.
You all know the central dogma, right? Information flows from DNA, gets transcribed into RNA, and then translated into protein. Well, transcriptional control is all about managing that very first arrow, the jump from DNA to RNA. If you can stop the process here, you save a ton of energy and resources.
The main players in this game are proteins called transcription factors, and they really are the gatekeepers of the genome. They basically come in 2 flavors, activators and repressors. Activators are like the go signal. They grab onto the DNA and essentially wave the RNA polymerase over, saying, hey, start copying right here. Repressors, on the other hand, are the stop signal. They clamp onto the DNA and physically block the polymerase, getting right in its way. It's a beautifully simple push-pull system that controls the flow of genetic information. And this diagram shows you exactly how they do it. It's a physical process. Up top, you see the activator protein binding to the DNA and helping to bring in the RNA polymerase, getting the whole process started. And down below, you see a repressor doing something a little more clever. It binds to the DNA and causes it to loop over, physically hiding the promoter from the polymerase. It's a direct, hands-on way of saying yes or no to making a gene. All right, now this is where the story gets really interesting. For decades, we kind of thought of RNA as just a boring messenger molecule, a simple courier. But it turns out, RNA is a massive player in its own right. So let's look at the layers of control that happen after the RNA has been made, where RNA itself starts calling the shots.
Now check out this incredible image. It shows something really special about bacteria. Because they don't have a nucleus separating their DNA from the rest of the cell, transcription and translation happen at the same time and in the same place. You can see the DNA strand running right through the middle, and peeling off it are the newly made messenger RNAs. And look, all those little black dots covering the mRNAs? Those are ribosomes, already translating the message into protein while the mRNA is still being copied. This tight coupling is a perfect setup for super fast, real-time regulation. And a major star of this post-transcriptional show is the small RNA, or sRNA. Now these guys are not destined to become proteins. Their whole job is to be regulators. They're short little pieces of RNA that go out and find a target messenger RNA, and then they bind to it. That simple act can either physically block the ribosome from getting on, or it can be a signal that says this message needs to be destroyed. They're like the system's nimble micromanagers.
So these sRNAs come in two main styles, which this diagram shows perfectly. On the left, in panel A, you have what are called cis-acting sRNAs. These are made from the exact same spot on the chromosome as their target, just on the opposite strand of DNA. This means they match up perfectly. Then on the right, in panel B, you've got the much more common trans-acting sRNAs. These are made somewhere else in the genome, and then they go hunting for their targets. Their binding isn't always perfect, which is actually a cool feature, because it allows one single sRNA to regulate a whole bunch of different mRNAs.
But maybe the most elegant piece of RNA regulation is the riboswitch. This is just brilliant. It's where the messenger RNA molecule itself acts as its own sensor. Part of the RNA folds into a very specific 3D shape that can directly bind a small molecule, like a vitamin or amino acid. When that molecule docks, it triggers the RNA to snap into a different shape. This change can then either stop transcription in its tracks or hide the spot where the ribosome needs to land. It completely cuts out the need for a protein sensor, making the response incredibly direct and incredibly fast.
And look, this isn't just some neat biological party trick. This has massive real-world importance. Take, for example, the pathogen Streptococcus pneumoniae. This is the bacterium behind nasty diseases like pneumonia and meningitis. And it turns out that these little RNA riboswitches are absolutely essential for its ability to make us sick.
And this chart says it all, really. What researchers did was compare the survival, the fitness, of normal bacteria with mutant bacteria that had broken riboswitches. The bar on the left is the normal wild-type bug, and you can see its fitness only drops a little bit during an infection. But look at the bar on the right. Those are the mutants with the broken riboswitches. Their fitness plummets. They just can't survive well inside a host. It's clear proof that these RNA regulators are a critical part of the bacterium's arsenal.
So we've decided to make a gene, the RNA has been transcribed, the ribosome's done its job. You'd think we'd be done, right? Not even close. There's one final layer of control, and it's all about modifying the finished protein product to tweak its activity on the fly. This is post-translational control.
You can think of this as the final fine-tuning. The protein is built, but now the cell can modify it. It can stick on chemical groups like a phosphate to flick an enzyme from off to on. Other small molecules can bind to the protein and change its shape, altering how well it works. The protein can be chopped up to activate it, or individual protein pieces can be assembled into a big functional machine. All of these tweaks are super fast, giving the cell an almost instantaneous way to change what its proteins are doing.
And the scale of this is just staggering. Take a look at this metabolic map of E. coli. Don't worry about reading every single pathway. Just look at all those little colored symbols next to the protein names. Each one, acetylation, phosphorylation, methylation, represents a different post-translational modification. It's a dizzying web of control that ensures the cell's metabolism is perfectly balanced and instantly responsive to any change.
Okay, so we've looked at all the individual pieces of the puzzle. Control at the DNA level, at the RNA level, and at the protein level. But the real power, the real genius, comes from how all of these layers are woven together into one seamless, integrated network. Let's pull back for the big picture.
This diagram does a fantastic job of tying everything together. It all starts with a signal from the outside world. That signal triggers control at the transcriptional level. But as that mRNA is being made, it's subject to its own regulation by things like riboswitches and small RNAs. Then, finally, after the protein is made, its activity is polished and fine-tuned by post-translational modifications. And you can see all the feedback loops. The end products feed back to control the beginning of the pathway. It's not a straight line. It's a living, breathing, dynamic network. So, what we're left with is this incredibly elegant, sophisticated system that bacteria have evolved over billions of years for one purpose: to survive. It's robust, it's efficient, and it's incredibly responsive. And that leads us to a final, really important question, especially for you as the next generation of biologists and bioengineers. As we try to build our own biological systems in the field of synthetic biology, what can we learn from these tiny masters? What secrets does this ancient blueprint hold for us as we try to engineer biology for ourselves?